Do we have a technology to edit our DNA precisely? Yes, now we have CRISPR-Cas9, a specific gene editing tool that helps us to modify, delete or correct precise regions of our DNA. Using this simple illustration by CB Insights, let us understand how CRISPR-Cas9 works within 5 minutes. Let's begin with what do you mean by this CRISPR and Cas9? This CRISPR means clustered, regularly interspaced short palindromic repeats. That means these are repeats that is present in the genome. Whereas Cas9 is a CRISPR-associated endonuclease that cut DNA at a location specified by a guide RNA. So it is simply an enzyme that cuts DNA. The system is discovered in bacteria. It is a part of bacterial defense system against viral attack. Now this system is considered as a versatile gene editing tool that revolutionizes medicine. Now let us see how CRISPR-Cas9 works. Let's take a simple example. Suppose this patient X has sickle cell disease that is caused by a single DNA letter mutation. We all know that sickle cell disease is a disease that is caused by a base pair substitution. So here it is CTC. This thymine is replaced by adenine. As a result, this glutamic acid is replaced by another amino acid, valine, that results in the formation of sickle cells. So first step is we'll be making a guide RNA. So scientists create guide RNA that matches to this target sequence. A guide RNA is a short piece of RNA that is complementary to the target DNA sequence. It is called as guide RNA as it guides Cas9 enzyme to the specific DNA sequence for cleavage or to make that specific cut. So if the sequence is known, scientists can prepare guide RNA that can specifically target specific DNA sequence or complementary DNA sequence. Next, this is complex with Cas9 enzyme. So Cas9 is a molecular scissor that can cut DNA molecule. So Cas9 and guide RNA forms a complex, Cas9 guide RNA complex, and that is used to correct this mutation. The cells of patient X receives this RNA Cas9 mix. This is what is actually happening. This guide RNA identifies the mutated DNA that causes sickle cell disease. Now clinical trials are going on. What is happening is from the patient we'll be isolating hematopoietic stem cell or progenitor cell with mutant beta globin gene, and that is causing the sickle cell red blood cells. So this is having this mutated T, and using CRISPR-Cas9 editing tool, we'll be replacing this T with A or adenine so that that mutation is nullified. As a result, this hematopoietic stem cells will become normal or wild type beta globin gene, and that can produce healthy red cells, and that is infused back or engraft back into the patient's bone marrow that can produce healthy red blood cells. So this Cas9 guide RNA complex is used to edit the genome of hematopoietic stem cells. So Cas9 cuts out that defective part or defective mutated DNA sequence guided by this guide RNA. Then this DNA repair system insert a healthy DNA sequence to replace that mutated sequence, or we will be providing a correct copy of the DNA, so it will be inserted by homologous recombination. If everything works fine, the patient X will be producing blood cells with wild type gene without that mutated DNA, so that patient no longer suffers from sickle cell disease. So this is how CRISPR-Cas9 works. Hopefully we'll be discussing the details of this CRISPR-Cas9 in the next video. Till then, take care, stay blessed. Thank you so much for your support. You are with biologicexams4u.com.